and we're a small company that does research on soil biology in most of Australia's crops. And today I'll be talking about the work I've done on sugarcane, which largely started in the mid-90s with the Sugar Yield Decline joint venture, and we'll continue for the next 3 years because I've got a new project on sugarcane where I'm going to be looking at some soil biology issues in sugarcane. And I guess what I'm planning to do in the next few minutes is just run through some of the main points I made in the talk that I gave in Mackay today. And I guess there's enormous interest in soil biology, but in my opinion there's not a lot of good understanding of what's really going on in soil. And so one of the first points I made is that a lot of the biological activity actually occurs on plant roots. Because what happens is that plants, they are taking energy from the sun and they're turning it into organic matter, but they're leaking some of that organic matter out of their roots. And we know from a scientific perspective that 20 or 30% of the carbon that plants produce actually leaks out of their roots as exudates. And you sort of got to question why that happens. Why would plants leak their carbon from their roots? And there's two reasons for that. What they want to do is create a biology around their roots that cycles nutrients and makes them available to plants. So the more organisms you've got in the root system, and the more things that are eating those microorganisms, the more nutrient cycling will happen, and the plant can actually utilise those nutrients. And the second reason is that there's a lot of pests and pathogens in soil that want to actually attack those roots and eat them. And so by having a biology around there, some of the antagonistic organisms actually help protect the plant from root diseases. So that's the reason that the roots is really the sort of cells where a lot of biological activity takes place. I guess the second point, and it really follows on from that, is that it's really carbon that's actually driving the soil biology. Carbon is an energy source for microorganisms, bacteria and fungi, for example. They decompose organic matter when it's produced, and it doesn't matter whether that organic matter comes from above ground material like leaves, it can be roots that are dying, it can be exudates out of roots. It doesn't really matter. Those bacteria and fungi will utilise that material, and then they're a food source for a whole lot of other organisms in the soil. All the bigger organisms then eat those bacteria and fungi, and then something even bigger comes along and eats them. So within the soil, what we call the soil food web, there's an enormous diversity of organisms, thousands and thousands of organisms, all interacting together and helping to not only cycle nutrients but control some of the pests that we have in our soils. I guess the third point that I made was that a lot of people think about when soil biology as how many bacteria and fungi are in the soil. And the problem with doing that is that these bacteria and fungi have life cycles of only hours or days, so the population can be something today and it'll be quite different tomorrow because there's been a rainfall event or something's changed. And so it's actually these larger organisms in soil that are actually a better indicator of what's really going on. So some of the nematodes, some of the arthropods they have life cycles in weeks or months, and so their populations don't change anywhere near as quickly as the fungi and bacteria. So there's fairly good evidence that if you have a look at these bigger organisms in the soil, you've got a fair idea of what the soil is doing, because if there's a lot of fungal feeding nematodes in soil, for example, it must tell you there's a lot of fungi in the soil. If you've got a lot of bacterial feeding nematodes, it tells you there's a lot of bacteria in soil. So it's often simpler and more easily interpretable if you actually measure some of the bigger organisms in soil when you're trying to get an indicator of what's going on in the soil. The fourth point I made was that there's really no silver bullet solutions to soil health problems. There's a lot of companies out there selling biological and organic products and claiming they have big effects, but there's not a lot of scientific evidence in a lot of cases that they actually work. And so I think growers need to be a bit careful when and certainly they can try these things, but if they want to try them, they ought to convince themselves they're actually getting value for money. And that means putting out some strip trials where they put the product out and have a comparison of no treatment, preferably 2 or 3 strips across a field, and actually measure whether they get a response in yield or do change the biology in some way. And I think then they'll be more convinced that they really are effective, or they say, oh, I'm wasting my money, I'll move on to something else. But in my opinion, we've spent far too much time in the last few years trying to look for those magic bullets when there's really some basic principles about how to manage soils to get a decent biology. And one of my final slides in this talk was about the 5 key principles for getting a decent soil biology. And the first is you've got to have a permanent residue cover or plant cover on the soil surface. So if you look at a natural system like a forest or a grassland, there's always organic matter on the soil surface, and that's driving the biology beneath it. So you always got to aim to keep that permanent cover. And in cane we produce a lot of organic matter. We have a trash blanket. But we don't want to see fallow periods where there's no organic matter there, because all the benefits you get from the trash are suddenly lost when you go into, say, a fallow period. The second thing is a diverse rotation sequence. If you grow single crop like sugarcane year after year after year, there's only a certain type of organic matter that's actually going into the soil. If you can add different types of organic matter, different bugs will colonise it, so you'll get a more diverse biology than you will if you have a single source of organic matter. So the more crops you can put into the system, so putting a legume is beneficial, but if you can put more than a legume, put some other crops in the system, we probably need evidence that it actually makes a difference, but the suggestion from the scientific literature is that it should make a difference and make things better. Organic matter inputs must be continuous, and I've just talked earlier about the fact that plants are the driving force, so you've got to have plants in the system all the time. Bare fallow, leaving land fallow is a disaster from a biological point of view, because all the organisms run out of something to feed on and tend to disappear from the system. The fourth one is reduction in tillage. We've been tilling our soils intensively for 100 years, and all that does is send carbon from the soil up into the atmosphere as carbon dioxide. And tillage also does a lot of bad things for the biology. It kills a lot of the big organisms. It tends to be the big organisms, the predators, that actually get killed by tillage because they just can't survive a rotary hoe or some sort of severe disturbance. So the more we can reduce tillage, the more likely we're to have a good range of biology. The problem, though, is in sugarcane that compaction is a major problem, and so people tend to till because they have to fix compaction. So you really got to solve the compaction problem if you're going to try and reduce tillage, and that's really a major issue the sugar industry must address, because compaction not only stops root growth. Now we know that when the soil is compact, roots can't grow through it, but it kills the habitat for a lot of these organisms. They need to live in pore spaces, and if you compact the soil so they've got no space to move, they'll die out. So a lot of the predators of things like nematodes have disappeared from our cane soils because we've squashed the soils to the point where those things can no longer live. So I guess the message is about managing the biology is if we can put those 5 practices together, then I think we'll make a start towards improving our soils, but it's not something that's going to happen overnight. It's something that we've taken 100 years to run down the biology of our soils and destroy all the carbon. We're not going to fix it in a few years. But by putting a farming system in place, it's got a chance to start to improve it. Then in the medium to long term, we should start to see some benefits.